Hi, I'm Colette Quinn, Microcalorimetry Product Manager at TA Instruments. I would like to welcome you and thank you for joining us in today's webinar. If you get disconnected at any time, please use the instructions you received to log back in. You can access numerous content by clicking the Documents icon in the bottom right-hand corner of your screen. This includes a speaker bio and additional file downloads. And if you need help at any time, click the question mark icon. Please ask any questions you may have at any time during the presentation by submitting them through the Q&A window. We will answer as many possible at the end of the webinar. Our presenter today is Professor Ernesto Friere. He is the Henry Walters Professor at Johns Hopkins University. He has been a member of the Department of Biology and Biophysics since 1986. He also holds a joint appointment in the Department of Biophysics and Biophysical Chemistry at Johns Hopkins Medical School. Since 2002, Professor Friere has been a scientific director at the Institute of Biocomputation, BIFI, University of Zaragoza, Spain. Professor Friere has pioneered the development of protein stability analysis and drug design strategies using thermodynamic and microcalorimetric techniques. The author or co-author of over 230 publications and several patents, Professor Friere is on several editorial boards. He has been the recipient of many awards, including the Hugh Huffman Memorial Award for pioneering contributions at the forefront of structural thermodynamics and drug discovery at the Calorimetry Conference in 2015. He has served on many scientific advisory committees for the National Institute of Health and the National Science Foundation. The title of his talk is Long-Term Stability Analysis of Monoclonal Antibodies by TAM Isothermal Calorimetry. Ernesto?
Thanks, Colette. So today I'm going to talk about the use of a new technology, the TAM isothermal calorimetry, on the analysis of long-term stability of monoclonal antibodies. As many of you know, biologics is the fastest growing segment in the pharmaceutical industry. It is expected that by 2018, about half of the drugs that are sold will be proteins, and the other half the conventional drugs. The extraordinary growth of biologics is seen also in this slide, in which you can see that the preclinical investment or value of biologics is about 3 times as much as those of small molecule drugs. So what are biologics? Biologics are protein drugs. In this slide, you can see the size comparison of an aspirin molecule, for example, that has a molecular weight of 180, with a monoclonal antibody, which is about 1,000 times larger. So what are the characteristics of protein drugs? Large molecular weight, as I said. All of them are produced by living organisms. They are expressed in bacteria, cell cultures, etc. They are very complex and have unique tertiary structures, and their function or activity is related to unique 3-dimensional structure. One extremely important characteristic of biologics is that they are not orally bioavailable. Traditionally, biologics were administered in hospital or clinical settings by IV injection to the patient. Nowadays, with the extraordinary growth of biologics, the tendency is to have the patient self-administer the drug. Since the dose is 1 or more milligrams per kilogram of weight of the patient, it is not unusual to have the need for 200 milligrams of protein to be injected. And for that amount to be self-administered in a sub-Q modality, you need a volume that is small, like 1 milliliter, which means that the concentration of the formulation will be on the order of 200 mg per mil, and that poses very important problems. Important problems like denaturation and aggregation that occurs at very high concentration. So there are two major goals in development, among others. One is to identify the monoclonal antibody that has the best denaturation aggregation profile, that is the most stable, and the one that has the smallest tendency to aggregate, and also to identify the buffer and excipient conditions that maximize the stability and minimize the aggregation of the biologic, or in most cases, the monoclonal antibody. Now, to give a short review of the standard situation with proteins, we know that when a protein is formulated, immediately after formulation, the vast majority of proteins will be in the native state, that is in the active conformation. But there are going to be few molecules in partially denatured states or denatured states. Any of these states may have a tendency to aggregate. If partially denatured states or denatured states have the tendency to aggregate, what we see in this slide is going to occur. At the beginning, the vast majority of molecules are in the native state, but those molecules that are denatured or partially denatured that aggregate will come out of solution and induce more molecules to denature or partially denature. And eventually, the amount of denatured and partially denatured protein will be high enough to make the protein solution useless from a therapeutic point of view. This is a process that occurs not in one day or in 1 week, but may take months or even years. So it is very important to select the antibody with the smallest tendency to denature and aggregate, the most stable, and also find the best buffer and excipient conditions that optimize those conditions. And from a technical point of view, it will be very important to have a technique that is able to identify the expected kinetics of denaturation of the antibodies over a long period of time. And this is where the TAM isothermal calorimeter is going to play a big role, I believe, in the development of biologics. So what is the TAM? The TAM is the first technology that measures directly the kinetics of denaturation aggregation by measuring the time evolution of the enthalpy of these processes. So the TAM, as all calorimeters, measure the heat that is released or absorbed by the process of denaturation aggregation. Now, all of you are familiar with the superior resolution of DSC, and now this resolution will be available or is available for isothermal measurements that last days, weeks, or months. So the time isothermal calorimetry provides critical answers to the issue of developing new monoclonal antibodies as protein drugs. One is the rapid determination of rates of denaturation and the rates of aggregation at constant temperature below the melting temperature, the TM, rapid ranking of different monoclonal antibodies and formulation buffers in terms of their denaturation aggregation kinetics, and a quantitative comparison of biosimilars and biobetters to the reference products. TAM isothermal calorimetry is truly orthogonal to existing techniques. It's not just one additional technique that measures temperature denaturation or chemical denaturation, etc. Let's recapitulate a little bit what the temperature denaturation of monoclonal antibodies look like. If a monoclonal antibody is allowed to undergo thermal denaturation, what is going to happen, besides looking at the nice peak that you see in DSC, is that the solution will become a total precipitated white milky stuff in which the antibody has been totally denatured and aggregated. Now, that process, not only for antibodies, but for many other proteins, is irreversible, and by being irreversible, it's not controlled by the equilibrium constant, but it's controlled by the rate constant. In this case, there are two rates that are going to be important: the rate of denaturation, Ku, how fast the antibody denatures, and the rate of aggregation, Kag, which is how fast the denatured or partially denatured state aggregate. Other situations may exist in which the native state will also have a tendency to aggregate. So what is the TAM? The TAM, or thermal activity monitor, measures the heat that is produced or absorbed, called heat flow, by a slow process that takes days, weeks, months at constant temperature. In the graph below, you can see the heat flow versus time in days for some arbitrary process at different temperatures. And this is what we are going to measure with monoclonal antibodies and other proteins. The operational temperature of the TAM is anywhere between 4 and 150 degrees, and the stability better than 10 to the minus 4, a tenth of a millidegree over 24 hours or more. And the sensitivity is better than 0.5 microcalories per second. The TAM consists of a big thermostat, and inside that thermostat that keeps the temperature constant, the calorimeter measuring modules are inserted. As you can see in the figure, these are 6 cell calorimeter modules, and those contain the measuring units and the calorimeter itself. Here you can see the inside of a calorimeter module. It has the sample ampoule that contains the protein, and also a reference cell that is sealed and that is used as to measure the differential heat between the protein sample and the reference. There are models that can contain up to 48 samples, and so it should be possible to measure many different antibodies or different formulations simultaneously. Now, these are just equations that explain the type of analysis that is made. As I said, we have 2 steps in this reaction. One is the denaturation, characterized by an enthalpy, and the other one, the aggregation, characterized by some heat of aggregation. And the total heat is going to be proportional to the amount of protein that has denatured plus the amount of protein that has been aggregated, each one multiplied by the amount of heat that they produce. And that can be written in terms of the rates Kag and Ku. And at the end, the quantity measured by the calorimeter is the heat flow, that is the rate at which the heat is produced or absorbed, and the last equation describes that process. So you can think of different scenarios that can occur. For example, if the denaturation is much faster than the aggregation, the 2 processes will be separated in time. You will have protein that is denatured, and after a while they will aggregate. Or if the aggregation constant is very large, or larger than the denaturation constant, then everything will occur in one step. Also, as I say, there could be native state aggregation, in which case something like that can occur. So how a TAM experiment proceeds. First, the instrument is equilibrated at the desired temperature below Tm. Measurements can be performed at formulation concentration. That's very important because not all techniques allow you to measure directly at the formulation concentration. That is 20 to more than 100 mg per mil of protein. Total volume of the sample, anywhere between 0.25 to 1 milliliter, even larger if desired. And it's the same as in a DSC experiment. The heat flow of the buffer is subtracted from the heat flow of the sample and then normalized by the amount of protein in the calorimeter cell. Now the next step is at what temperature we start our experiments. And for that we use DSC scan of the sample as a guide to select the temperature for the initial experiment. So here's a characteristic times of denaturation below TM that can be used as a guide to set up an experiment. For example, if we have a protein that melts with a TM of 82.5 degrees, if we measure at 70 degrees, that is 12 degrees below TM, the denaturation aggregation will take about 8 hours. It will do it at 65 degrees. It will take 55 hours, and so on and so forth. So for our studies, we usually start our experiments 12 degrees below TM to have an idea of what the rates are at those temperatures, rather than using a very, very low temperature that will take weeks or months to occur. So, for example, this is a DSC of lysozyme, which we have used as a test protein to set up the technique and the technology at pH 9. As you can see, under those conditions, it denatured at 73.5 degrees, and after a while it aggregates and precipitates, as you can see by the exothermic peak after the TM. So we start experiments at 59 degrees with the TAM, and this is what we observe. So this is the heat flow associated with lysozyme, pH 9, 59 degrees, as a function of days. And you can see that at the beginning there is a endothermic heat flow, and after a while it comes down to 0, and then that endothermic heat flow is followed by an exothermic. That's a signature of denaturation preceding aggregation. That is, the endothermic peak associated with denaturation occurs before than the exothermic peak associated with aggregation. If we do those experiments at different temperatures, the TAM will produce something like that. And on the right-hand side, I have the rate constants that were calculated for aggregation, that were measured for aggregation and denaturation at the 3 different temperatures. And more importantly, the TAM experiments allow you to calculate the percent of denature and percent of aggregated protein at the different temperatures. In the left graph, you see in blue, the percent of denatured protein, and in red, the percent of aggregated protein, which occurs after denaturation, as expected from the experiment. And on the right-hand side, the same graph, but at 59 degrees. You can see that at lower temperature, there is a wider separation between the percent of protein that denatures and the percent of protein that aggregates, whereas at high temperature, at some point, everything will converge and denature closer together. So the TAM isothermal calorimetry allows separation of the kinetics of denaturation and aggregation, and this is important information to develop rational strategies for formulation optimization. You know which process is faster, which process is causing problems, and then you know exactly whether to stabilize or prevent aggregation is a priority. These are now monoclonal antibodies, and for many monoclonal antibodies, denaturation and aggregation occur simultaneously, and therefore no separate endotherm and exotherms are present. Everything occurs simultaneously, and the data is well accounted by a single exponential, as you can see in the figure. As in the previous case, it is also possible to calculate the percent of denature aggregated, in this case occurring at the same time, as a function of time, which we are expressing in terms of days. We have an example of native state aggregation. In this case, as you can see in the graph, the heat is exotherm because there is aggregation with no denaturation, and it starts exothermic until it goes to 0 and the protein is aggregated. In this case also, we can calculate the percent of protein that has been aggregated as a function of time in the days of incubation. So I'm going to present now two examples for two different antibodies. The first one will be a temperature study, and the second one will be a study in which we determine which one is the best formulation. Now, this is the Dcf of monoclonal antibody Y. You can see it's characterized by two peaks, a small peak at the beginning that is usually attributed to CH2 domains, and followed by the main peak of denaturation. In many cases, as in this case, the first small peak in the calorimetry is reversible. That is, if you heat the protein up to 75 degrees and you cool it, that peak still is present. And the TAM is going to be blind to that reversible transition because there is not an active process of heat production as a function of time. It's an equilibrium situation. And therefore, in calculating the temperatures to start the TAM experiment, we consider the main peak and do experiments in this case at 73, 70, and 66 degrees. The main transition was 82.5 degrees centigrade. Here on the left graph is the heat flow as a function of the number of days of incubation at the different temperatures studied. And on the right-hand side is are the rates obtained at the three different temperatures in terms of days to the -1. And these experiments were performed at 100 mg per mil, which is a typical formulation concentration. These are the rates as a function of temperature. And as before, it is possible to calculate from the data what is the percentage of denature aggregated protein as a function of days at different temperatures. And that's one of the most important features of the TAM that it will allow you to determine not only the rates, but from the rates calculate how much protein is aggregated, how much protein is denatured at different temperatures or under different formulation condition, which is what I'm going to describe now. So this is a monoclonal antibody different than the previous one. The denaturation is characterized by a single peak at about 71 degrees. We started measuring at 60 degrees, which is about 11 degrees below TM. And this is the heat flow as a function of time. And as you can see, as in the previous cases, no separate endotherm and exotherm are visible, and the data is accounted for by a single exponential. Now, if we run the same experiment with different buffers and different excipients, that is, different formulation conditions, we see results like the ones shown in this graph. We have a buffer condition or formulation condition in which denaturation aggregation occurs very fast, in about half a day, and at 60 degrees, and conditions in which denaturation aggregation occurs very slowly, about 3 days. So this type of experiment will allow you to select which buffers give you the best denaturation aggregation profile. These are rates that were determined with different formulations, 4 different formulations. These are the percent of denatured and aggregated protein in the different buffer conditions as a function of the time in days. And you can see right away that buffer A is the worst, buffer D is the better, because denaturation aggregation occurred much more slowly. In this particular example, size exclusion chromatography experiments were available for the 4 different formulations. And we took the data after 10 weeks. And if we plotted the percent of aggregates after 10 weeks incubation at 25 degrees, we can see that they correlate very well with the rates of denaturation aggregation obtained by the TAM. So, to summarize, TAM is capable of quantitatively ranking different monoclonal antibodies and formulation buffers in terms of rates of denaturation aggregation, and the rates that are obtained by TAM can be gathered in a very few days and are correlated with size exclusion chromatography very well, and data that is obtained over several weeks or months. And to conclude, TAM is a promising new technology that will help accelerate the development of biologics. Thank you very much.
Thank you, Ernesto, for that informative presentation. A recorded version of this webinar will be archived and available on the TA Instruments website. We will now begin the questions and answers segment of this webinar. Please submit your questions using the Q&A window. We'll do our best to answer as many as possible.
So we have a first question coming in here. And so Professor Friere, one of the things that you mentioned is that you do a buffer scan. Have you had any insight into, unstable or excipients that would have been added, such as polysorbates or reducing agents that helped someone with the buffer formulation? Or has this been just mainly taking the buffers as is?
Well, in the experiment that I mentioned, by buffer we meant buffers with different excipients that unfortunately we are not allowed to tell what exactly they are. And most of the industry
proprietary. Okay.
Yep. But by buffers there are combinations of different buffers and excipients and salt and etc.
Okay, but I mean, I'll address that. So theoretically, if you do have an unstable excipient in your buffer, we would anticipate that you'd be able to see a heat flow event associated with that event. For example, if you have a reducing agent that is oxidizing over time.
Yeah, always.
Events, right? Right.
Yeah, obviously, if you have a reagent that is going to degrade, you're going to see it, but obviously the excipients that are used in formulation for clinical use should be stable.
Right. Okay, so it'd be mainly like an earlier step. Okay. Let's see. So you did bring up so another question coming in from somebody else. So it says, so you brought up the possibility of an antibody going from its native state to aggregation. Have you encountered any of those situations in the samples that you've assayed so far? And can you make any comments to that kind of situation and what the thermogram looked like?
Well, there is one example given in the webinar, which is the case in which we see the antibody aggregating in the native state. You see an exothermic peak. Since then, we have seen many other situations, situations in which the antibody aggregates in the native state and later it denatures. So the TAM allows you to not only see the two processes, but which process is the one that occurs at the faster rate. And you can take, I mean, you know what the origin of the instability is, either aggregation directly or denaturation that triggers, or partial denaturation that triggers aggregation.
Okay. So I'm just waiting a few more coming in here. Okay, so so a lot of these studies were done at an elevated temperature. And you do mention that you could potentially do some studies at a lower temperature. It would just take a longer period of time. Have you attempted any of those assays of doing a low temp study, for example, studies at the administration temperature? And then if you have not, what are the reasons? Is it, yeah, so that's just, so have you done any studies at the administration temperature? And if so, what were the timelines like?
No, we haven't because they will take months, and obviously the main goal is to try to have a very quick identification of either the antibody or the solution conditions that guarantee a maximal stability. So if you do the experiment, let's say at 40 degrees or 25 degrees, it will take a year or so to complete. And in that case, you can just as well use size exclusion chromatography and have directly measurement the amount of monomer or high molecular weight aggregate. So in principle, you can do the experiment, but I don't see what the point will be.
Right, because you've already proven in one of your slides with the size exclusion chromatography data at 25 matching up very nicely with your elevated temp studies. Okay. Okay, so you mentioned some volumes in here that were used in this assay of anywhere from 250 microliter to 1 ml. What was the most common volumes and concentrations that you found that have worked well for this type of assay?
In the experiment that we have performed, and our collaborators at the formulation stage usually have vast amounts of antibodies. We have used 1 milliliter or 0.5 milliliters, and concentrations ranging from 25 mg per mil to 100 mg per mil. That is, we can measure directly with the TAM the stability of the antibody at the required concentration, because even in size exclusion chromatography, you can incubate a sample at high concentration, but then you have to dilute it to run the experiment. And if you have a aggregates that dissociate, they won't show up.
Okay. So it's usually the concentrations aren't limited by the technique, it's more that the concentrations you needed to run those assays there because they were requested. Okay. All right.
Well, here we are not running experiments in trying to see what is the lowest concentration that we can measure. I mean, people in the pharmaceutical industry want to know what happens at 100 mg per mil, and that's what we are doing.
Okay. So it looks like we just have, we'll make time for just one more question, and I'll go through here and figure out which one we'll do. All right.
Well,
so,
let me comment on the previous question, because aggregation is a concentration-dependent process. And if a formulation of an antibody is going to be 100 mg per mil, you need to know what happens at 100 mg per mil. Unfortunately, not all the techniques are capable of measuring directly at such a high concentration. And that's something that the TAM addresses perfectly well. Yes, what was the other
So we'll just
question?
end it up with one more, and that is in particular to the question of aggregation. So have you considered any ancillary techniques for measuring aggregation to, that will correspond with your studies that you have found here, the initial results?
Yes, obviously the most widely used technique is size exclusion chromatography, and that's usually our collaborators have performed that, and that's what we are using to correlate. But also light scattering will be a technique that will correlate very well with the data from the TAM, and we expect to have light scattering data in the future and obtain side by side with the TAM data.
Okay. Well, those are all the questions that we have time for during this live session. But please go ahead and continue to submit your questions, and we can address those either myself or Professor Friere via email. And I just want to thank everybody for logging on today and listening to our webinar.